Today, I had the opportunity to speak with Dr. Mathews from the Levin Lab, where she works at the cutting edge of bioelectricity. We covered a wide range of fascinating topics from optogenetics and bioelectric signaling to the role of mitochondria in cancerous states. We also touched on emerging areas like biophotons, liquid crystals, and how biological systems adapt through processes like habituation and sensitization. Because Dr. Matthews is a busy researcher and had an experiment to get back to, this conversation moves quickly, and you may notice a few visual and audio inconsistencies from recording in an active lab environment. But that being said, there are some real gems throughout. I'd love to hear what stood out to you and what questions you'd want to explore with Dr. Matthews in a future conversation. Well, Dr. Matthews, thank you so much for joining us here today. Before we get into the science itself, I'd like to start with your journey into this field, perhaps what inspired you, a bit about your educational background, and how you ended up working in the Levin Lab.
I have always wanted to do genetic engineering. I think I probably told my dad that I wanted to be a genetic engineer when I was about 9 years old. I grew up in uh, a home that was full of engineers. So my grandfather was a mining engineer, but he also did, electronics, geology, all sorts of different things. My dad is a geologist, but also a civil engineer. They have always built things. I grew up in a lab that had machining equipment, electronics, a chemical lab, everything in it. So I always told them that you guys work with rocks and I want to work with cells. So that's how I grew up. And then when I was 16 years old, my uncle gave me a book called The Bioelectric. And I started reading that book, and I was really inspired by that book. And then I told my high school science teacher that I wanted to do an independent project based on salamander regeneration in an electromagnetic field. So my dad helped me build an electromagnetic coil. So we sat there and actually coiled, all of the copper wire around a large cylinder, and we made it so that it would be the right intensity to kind of recapitulate what Robert Becker had done in his Bioelectric book. And my professor got me some salamanders, and I put the salamanders in there after amputating their legs and saw whether or not they regenerated faster. So that was my independent project for high school. Um, and so I really wanted to work on that. I found that fascinating, but nobody was really working on it at that time. Becker's stuff had kind of gone to the wayside. And a lot of that was because kind of the meshing of electrical engineering with biology is very difficult because you have two people with very different, uh, you know, backgrounds and talent sets. There's not a lot of electrical engineers that understand much about biology, and there's not a lot of biologists that understand electrical engineering. You don't take the same course requirements, right? So the people that were really working on it at that time were people that were looking at high uh, high power electrical lines and seeing if there was any sort of connection between like leukemia clusters and these really high intensity electrical lines. And that wasn't exactly what I was looking for. I was looking for something a little bit more mechanistic on the regeneration side. And so I said to myself, well, the only person that's going to really like bridge all of this is a biochemist, because I think you really need to explain what's happening using these electromagnetic fields in a biochemical language so that the scientists can understand it, the biologists can understand it. And so I went and I got my degree in biochemistry at the University of Nevada, Reno. I grew up in Nevada. And then afterwards, I couldn't find any sort of programs that were, again, doing this sort of stuff. Mike had not come out on the scene at that point. And so I said, okay, well, I'll do the next best thing. I'll work on anti aging research, which I thought was pretty interesting. And so I started working for Sierra Sciences in Nevada. And I started out as a lab admin, and then I became a research technician. And then I fell in love with this guy in Oregon, and I moved to Portland, Oregon. I went and I worked at OHSU for a little while on uh, uh, basically, different neurological disorders, ALS. And I didn't like doing the mouse studies and having to kill little uh, mouse pups with horrible chemicals and then find out what kind of lesions they had on their brains. And so then I also worked over in Corvallis, and I worked on anti-bioterrorism research. So trying to come up with new ways of combating anthrax, smallpox, things like that. And then I'm keeping my eyes out this whole time. I'm still researching. I'm still collecting papers on regeneration and seeing what's out there. And then I just couldn't find anything, and I was like, okay, well, what's next? What would I like to do? And so I thought sustainability was really important. So I went and I went to the University of Hawaii in Manoa, and my project was metabolic engineering of E. coli bacteria for biohydrogen production. So that was kind of cool because I got to do what I wanted to always do, which was genetic engineering. So I got to, cut out certain metabolic switches in bacteria and replace them with certain engineered hydrogenases that we were working with. And so then during that time, I was starting to see Mike's name kind of coming up. And I said, well, hey, this is the guy. This is what I want to do. And so I reached out to him and I told him, I'd really like to do a postdoc in your lab. And he said, well, I don't, I just started my lab. I don't have any space for someone right now. And I said, well, what do you need? What kind of background do you need? And he's like, well, I'd really like someone to take on mammalian cells and apply some of the stuff that we've been doing with the tadpoles into mammalian systems. And so I went and I said, okay, well, I'll get those skills and I'll come knocking on your door later. And so yeah, I did a postdoc over at UH as well for the Institute for Biogenesis Research, and I got to do basically the same thing that I had been doing with bacteria, but doing it on mammalian cells. So we were genetically engineering them. We were reprogramming human adipose cells into stem cells and then driving them into a cardiac lineage. And so the whole idea was trying to use stem cell therapy to help people that had heart attacks and to regrow those areas. So I did that for 2 years, and I told Mike, well, now I know how to edit mammalian cells. I got all of this broad cell culture experience, and can I come in and interview with you? And so, come to find out, Mike actually got put on the same track with the same book and around the same age. But he's 10 years older than me, so he got a 10-year head start. But yeah, I felt like I was at home. It was funny. I gave the talk for the interview, and he said, I probably would have given an extremely similar talk. I was like, yeah. So I knew I was in the right place.
And it's a really good book for me that acted as a gateway as well into the regeneration and bioelectricity and just that whole world. So I highly recommend if someone hasn't read it to do so. So it seems like, in terms of talking about regeneration, that bioelectricity is playing a big role. Could you talk a little bit about what the role might be and what do we mean when we refer to bioelectricity? Where does it come from? A bit of information about that.
Yeah, so bioelectricity, in the sense that we're using it, is a we're very interested in resting membrane potential. And so, what is that? Well, all cells have a resting membrane potential, and all that is the balance of positive and negative charges inside and outside of the cell. So we call a cell hyperpolarized, meaning that it is more negative. So it's more strongly polarized from the outside, and that is a cell that if you look at the resting membrane potential signatures of all cells, hyperpolarized cells tend to be cells that have terminally differentiated. They are your neurons, your muscle cells, anything that has, is no longer dividing, actively dividing. And then there's cells that are kind of in the middle. So, like your liver cells are somewhat in the middle. I would say hyperpolarized cells are around negative 80 millivolts, their normal resting membrane potential. Liver cells are around negative 30, negative 40 millivolts. So they have some regenerative capability. And then there's your depolarized cells, and those are the ones that are less negative. I always like to tell people that when you're young, you're less negative. When you get older, you get more negative, right? You get more hyperpolarized, and so embryonic cells are very depolarized. Cancer cells are very depolarized. Anything that's actively dividing is very depolarized. And so that's what we mean by bioelectricity in the sense that we use it most of the time. Now, there are also active bioelectrical signals that are being sent between cells, and that's what a lot of neuroscientists and people that study the heart or muscle are looking at. They're looking at action potentials, and those are very large depolarizations that occur at certain frequencies, right? But a lot of the regeneration stuff that we're looking at seems to be more about resting membrane potential. And so it seems that the resting membrane potential of a cell kind of sets the stage for what the transcriptional outlook is going to be and epigenetic outlook is going to be for that cell. So some of the experiments that Mike has done in the past has been to induce whole organ formations in areas of a tadpole where they're not supposed to form, right? So one of the things that he did was he got an eye to be induced on the tail of a tadpole, and that eye actually formed an optic nerve. It went and traveled to the brain, and then when that frog grew up, it could actually see out of that eye. And that is not possible with transcription factors alone. So I believe it's PAX6. You can express PAX6 in a general head area, and then you can get eyes to form in wherever you're expressing that gene. But you cannot get eyes to form in the tail area when you use PAX6. And so, what you can look at as depolarization and hyperpolarization is setting the stage of those areas that are then going to be able to become those organs. So the tail in a tadpole is more hyperpolarized than the head, and so the head is more depolarized. And so if you can then take that tail area and depolarize it a bit, then you can get the induction of that. Now, that doesn't mean that we depolarized that area and then added PAX6. That's not what we did. We actually just depolarized that area. And it wasn't like every single time you get an eye. Sometimes you get different organs. It really depends. You have to be on the right area. You have to hit the right number, right? Like, so negative 30, let's say you get an eye. That's not correct, but I'm just giving you an example. Negative 30, you get an eye. Maybe, negative 50, you get a heart, or, negative 20, you get a limb, right? So each resting membrane potential, probably the duration of that resting membrane potential as well, is going to change that epigenetic programming of that area in development. And so then he went on to do some stuff with regeneration as well. And so during regeneration, what you get is a depolarization of the area that is being cut off or amputated. So wherever you have that kind of bolus of cells that is now going to become the limb. And so what ends up happening is that if the animal is at a refractory stage where it's no longer, regenerating, that electrical signature doesn't stay for as long as it needs to in order for that regeneration process to occur. And so what Mike has done is kind of given it a little kick, right? And he can do that with drugs. He can do that with optogenetics, which is kind of something that neuroscientists use, where you can shine a light and an ion channel opens during the time that you shine off that light, and then that will reset that resting membrane potential. And he doesn't have to do it for very long. All you need to do is to give it that kick, and then once you've given that kick, the whole program will start. So it's really nice because you don't have to micromanage things. You're basically just giving the master instruction of this is what's supposed to be in here, and then the rest of it kind of follows. And I think that's fascinating. That's a really cool level of control.
Listening to things about bioelectricity, I've heard the term cognitive glue come up a bunch. What does that refer to exactly?
So that's more the signaling that's going on. And so then the idea is that all cells are communicating bioelectrically, and they're not communicating with these large potentials, right? They're communicating with kind of these more sub-threshold changes and potentials. And so, um, you know, Mike strongly says, that all cells have the same type of properties as neurons, but they're just not as specialized, right? And so they have these electrical communications that are occurring with one another, and that instead of having cognition or thought come out of it like it does with neurons, his idea is that morphology and where the cells are supposed to be and what structures they're supposed to create is what that information is that's being passed along between those cells. But they are coordinating. So a lot of the stuff that we do in this lab, if you look at the papers that come out of our lab, you're like, oh my God, like this is like 15 different labs. But the overarching theme is, how do units come together and create a system, right? And how do they communicate with one another? So he does that computationally. We do it with cells. We do it by kind of perturbing developmental systems and saying, okay, let's take these cells out of this system and we make something new with it. How does it form? How does it coordinate? Like, where do the instructions come from with collectives of cells that are no longer coupled to the larger organism? What do they do, right? Which is also really, really great, because a lot of people don't go outside of that box, right? They just stay in that developmental biology box, or mechanistic box. And so I think we learn a lot about how cells organize themselves, and even just units and systems as a whole, right? And how do you use that in just trying to compute problems better? So we've also been doing a lot of stuff in AI as well, and looking at neural networks, looking at different computational pathways that are going to be more effective than what we have, just by trying to mimic how cells use this non-localized, spread out computational method of talking to one another.
What happens when that starts to break down? I'm not sure if there's a term for the opposite of the cognitive glue, but I'm kind of wondering, is that what leads to a cancerous state?
Yeah, so I just submitted a paper. It's in a preprint right now, but um, basically, cancer seems to be that, right? It's a disorder of order, right? Like it's a disorder of being part of that whole. And what it seems, the steps that are involved in that is that if you get some sort of injury, or if you get some toxic insult, uh, you know, what happens at that point is you get inflammation in that tissue area. And inflammation causes the release of reactive oxygen species in the tissue. And the release of those reactive oxygen species actually shuts down the communication between the cells. It closes what we call gap junctions. And gap junctions are like little holes that, connect the cells up to one another and allow them to share information. And so you get ions going in between gap junctions, you get small molecules like serotonin go between gap junctions, but it is restricted, so you don't get a lot of proteins, large proteins going through gap junctions. But that gap junctional communication really has an effect on the resting membrane potential of the cells. And so when all of those cells are really well coupled, they're actually more hyperpolarized. They're sharing the charge between one another. And then when these ROSs come up, these gap junctions can then, and at first, cells like to share stress among other cells to see if they can normalize it, right? Like, are there enough of us that we can maybe make this cell like apoptose or senescent, and then it's gone. But if it's a large enough area, right, you have enough reactive oxygen species in that area, then the collective can't dampen down that ROS signal, and then those ROSs shut down the communication between those cells and the other outside cells. And once that happens, those cells depolarize. And when they depolarize, now they're more able to proliferate, right? So then now you get the proliferation occurring. And proliferation uh, in the presence of reactive oxygen species is what causes mutation, right? And so that's when you start to get all the problems, mitochondrial problems because of the metabolism and the reactive oxygen species. Mitochondrial problems can cause genome instability, epigenetic changes because of those inflammation markers, and because they're not being shared with everything else, and you have this highly proliferative environment, then cancer starts to take off, right?
Yeah, I was surprised. I was listening to a video, it might have been a month ago, and you were a part of it in a collaboration talking about the nuclear mitochondrial transfer experiment. For those who may be unaware of it, could you explain what it is? And I guess for me personally, I had no idea the role that mitochondria play or might play in terms of cancer.
Yeah, so I just had a talk with, I can't remember his name, but he's a leading researcher in metabolism and cancer. And basically, he says that there is absolutely no genetic component to it, that that's all a lie, that it is primarily driven by mitochondria. And so you can take the nucleus of a cancer cell and you can put that into a healthy cell and it will not become cancerous. But then you can take the mitochondria of a cancerous cell and replace the mitochondria of a healthy cell with those, and it will become cancerous. And so it seems that the mitochondria themselves get so reprogrammed with things that they are really driving this cancer. And so it looks like the waste products that are being created by the mitochondria, because mitochondria are the ones that are driving ROS production, and then they're also driving a lot of like the acidic byproducts and things that are happening in the cell, and determining ATP ADP ratios, NADH NAD plus ratios, and then all of that has downstream effects. And then here's something else that's really crazy is that the mitochondria have their own ion channels. And so, we there's a paper that soon will be published. I don't know if it's pre-printed or not, by Servio Gentile's group. And he was saying that the drug that they were looking at that was opening up a particular potassium channel, they thought that it was working on the outside membrane potassium channel, and that's how it was working. And they found out that it wasn't. It was actually working on the mitochondrial potassium channel. So, a lot of these different organs, organelles, so the ER, endoplasmic reticulum. It also has ion channels. The mitochondria has ion channels. There's a lot of places where these ion channels are kind of coming into play that aren't looked at traditionally, right? And it's a lot of it is difficult. It's difficult to look at those ion channels. It's already difficult to look at them in the plasma membrane, isolating mitochondria and trying to look at their ion channels in any sense that would be translatable to what you actually see in the dish. Is going to be extra difficult, but that's where a lot of these new dyes and also genetically encoded voltage indicators that you can target to particular organelles and stuff. That's where that's going to come in and really open up that field. And it's starting to.
It sounds like we're almost going to need mitochondrial transplants in the future to fix a cell or something like that. I'm curious how all of this changes the extracellular environment. We have these changes going on inside the cell. Does the extracellular environment stay normal or does it change as well?
No, it stiffens up. So what happens is reactive oxygen species can affect the way the cross-linking occurs in the extracellular, the ECM, so the extracellular matrix, and it makes things stiffer. And that feeds into the whole depolarization because there are ion channels on cells that are they're mechanically sensitive ion channels. So in response to stiffer environments, they will allow more of a certain ion channel in. They're called TERP channels. And those are depolarizing when you're in a stiff environment. And so there was actually a really great paper that came out where they were giving mice like a breast cancer, and what they did is they just used fascia pulling on the area where the tumor was growing, and they saw that they had a reduction of like I think it was 30 or 40% of the tumor growth just due to that manipulation because they weren't allowing it to stiffen up to create even more of a feedback mechanism that would depolarize that area. And the same thing occurs too, like when you have cancer cells. Cancer cells will release what's called TGF beta and some other cytokines that then reprogram the fibroblasts that are in the tumor microenvironment to become what they call cancer associated fibroblasts. And cancer associated fibroblasts will change the collagen linking in the extracellular environment, also making it stiffer. And they will also act as the cloaking device for the tumor. So they are the ones that are stopping the immune system from recognizing the tumor for what it is. And because of the cytokines that they're releasing, basically block those immune cells from being activated in that area.
A bit ago, you had mentioned senescent cells. It's one of those things that I often hear, but don't know that much about. So, what are they, and do they play a role in cancer at all?
Yeah, so senescent cells, in and of themselves, can also be drivers for making cancer worse. Senescent cells will give off what's called a SASP signaling, and that is kind of a cocktail of different cytokines. And that SASP signaling can drive the growth of tumors, and it can also help cloak those tumors as well. And so there's a thought that the reason why, and it drives inflammation. So senescent cells drive inflammation. So thought is that as we get older, one of the reasons that we start to develop more cancer is because we have a buildup of senescent cells, and those senescent cells aren't getting cleared out. And so then they create this inflammatory environment because they're releasing these cytokines. And then that inflammatory environment is then what drives cancer and all that. So, one of the big anti-aging pushes is to come up with senolytics, which are drugs that will clear out these senescent cells. And what senescent cells basically are is, if you take, let's say you take some endothelial cells, like the lining of the gut, and you put them into a dish, And this isn't a cell line. These are what we call primary cells. You're grabbing them right from the body. You're not doing anything weird to change them. They will eventually become what's called senescent because they have reached the end of their division. Their telomeres are short now. They're no longer able to divide, and so they sit there. And the you can always tell senescent cells, they usually get really large, and sometimes their nuclei kind of fragment inside of the cell, so you get like multiple little nuclei. And so then what's supposed to come in and clear all of that out is autophagy, right? So that's what fasting and everything else is supposed to help put your body into, is a state of autophagy, and then you can kind of clear out these dead cells. Well, not dead cells, but malfunctioning cells, right? Your body just eats them up, and then recycles all of the nutrients and stuff, and builds new cells. But when you're constantly eating high density nutrient rich foods, and you never give your body a chance to actually clear that out, then that process doesn't occur. So you get a buildup.
One thing that stood out to me as I was looking through your papers as well as an interview I saw is how well versed you are in so many topics and just your interests. And in one, you had mentioned optogenetics and some work that you were doing in that. I was wondering if you could talk a bit about that and what your thoughts are.
Yeah, so optogenetics is really it's fascinating. They're using it primarily in neuroscience. So you shine a light, usually a blue light, and then it opens up an ion channel. And this was actually based on ion channels that were in algae. And so the algae, when they are exposed to light in the ocean, this is ocean algae, then they would open up these certain ion channels, and then they could photosynthesize and all this other stuff. So the ion channels have been modified now so that you can do very fast pulses. So you can use very fast pulses of light and get very quick action potentials. And then you can see whether or not you can optogenetically create a certain behavior in an animal. And so they will, put these light fiber optics into the brain of these mice that have had these optogenetic channels put into them, usually using a viral vector, and then they're able to manipulate their behavior by kind of writing in the different patterns that they see during other behaviors that they're reading out, right? So this whole idea of reading out and writing in is something that has really fascinated Mike. And so in neuroscience, we read out usually with calcium transients as kind of a proxy for a voltage change. And you can also read out with what's called genetically encoded voltage indicators, which aren't as sensitive as our genetically encoded calcium indicators. But we find that in non-neuronal cells, calcium transients don't have a one-to-one mapping with voltage transients. So we use those types of things to kind of read out what's happening in collective of cells. And then we want to use optogenetics to kind of write in certain patterns. And what does that do to, like, let's say, the proliferation of cancer if we impose the voltage patterns that we see in normal cells, right? Or what does it do to normal cells if we impose the voltage patterns that we see in cancerous cells, right? So we can kind of think about those things. The other thing, just besides the regular transients, is that we want to try to use optogenetics to manipulate the resting membrane potential. And that's a lot harder because optogenetics were built to create very fast transients. And resting membrane potential, you really have to hold that ion channel open. And so traditionally, you would have to have that blue light on the whole time. And cells can experience what's called phototoxicity, where they just can't handle that amount of light. It'll kill them. It builds up reactive oxygen species, and then they basically die. So what I've been doing is really delving into all of the tools that all of these people that were developing them didn't want, right? Like, what are your slowest channels? What were the mutations that you had where you saw that those channels were open for a lot longer? And so there are some channels. Blink 2 is one that's it's a potassium hyperpolarization channel. You can shine the light on it for 3 minutes. That opens up the channel completely, and then it takes 10 minutes for it to close completely. So you can really modulate that membrane potential. So I've been kind of messing around with doing some mutations in other ion channels to try to get them to stay open for longer. And we're in the process right now of kind of doing the quantification and the qualification of like how well those are working.
That's excellent. I'll have to keep an eye out for that. In terms of light that the cell is already making, biophotons, have you heard anything about how it affects microtubules, perhaps as they may act as light guides, or even biophotons being used in DNA repair?
Yeah, so there's, biophotons is, I love that subject. So the way that biophotons work is that you have your mitochondria, it's giving off reactive oxygen species. The mitochondria kind of sit on the microtubules and they get shuttled up and down those microtubules and they go like from the periphery over to the nucleus and back. So they're always being moved around. And what's interesting is that reactive oxygen species, once it gets released from the mitochondria, it will hit the side of the microtubule. And when it does that, the inside of the microtubule releases a biophoton. And then that's what travels through the microtubules. So the microtubules are acting as like the fiber optic cables into that. Now, the microtubules are also being attached to the nucleus of the cell, right? And so there's some excellent work that has shown that DNA actually acts as a light guide itself. And so these biophotons are going into the DNA, and as they travel through the DNA, there are little proteins on the outside of the DNA that act as kind of the alert switches that there might be a break in the circuit. So if you have a mutation, let's say like a missense between pairs on the DNA, then the light won't travel the right way, and the switch on the outside of the proteins that are kind of acting as like these little, guards or what do you call them? Outposts, right? So they'll flip and they'll change their conformation. And when that happens, all of the DNA repair machinery goes to that area and starts repairing. And there, I'm so horrible with names, but there's a really excellent researcher that found that that was actually the case, and so she calls them DNA wires. And you can see that she has shown that it does occur, that the light is traveling through them. And then she's the one that proposed that that was the main way that you would have DNA repair occurring so quickly because the light's so fast, right? And then the conformational change is that what actually gets those complexes in there to fix it. So the idea is if your mitochondrial health is bad, if those reactive those reactive oxygen species are not directing those photonic waves in the correct way, then your DNA is no longer able to correct its problems that it has because it's not getting the right pulses through it, right? And so that DNA repair machinery slows down and you get a buildup of mutations and whatever else.
I guess on that note, I'm curious because based on what I've read, we could consider DNA and microtubules to be a sort of liquid crystal. So, do you think its ability to interact with light is related to its highly ordered state? And if so, would other things that are categorized that way also probably have similar properties?
Oh yeah, so there's a really great book. It's called Cells, Gels, and the Engines of Life, and it talks about you know there not being this whole idea that the cell is like this sack of water and everything's just kind of floating around in it is completely wrong, right? So we need to really think of the cell as being like Jello, but ordered Jello, right? And that there's ordered water that's inside of the cell, and that water, instead of just bouncing all over the place, is kind of locked in because it's ordered on all of the different surfaces of all the charged molecules that are there. And then that particular order that's right next to those molecules is also exerting order on the other water molecules that aren't particularly, and I don't remember how many layers it can go, but that light itself can act to increase that order or decrease that order. And so I believe they showed that when you shine infrared light on water molecules, that they will stack up and form this ordered molecule, stacking in more layers, right, than you would if you don't have that light. So I think that light is probably driving a lot of things. The other thing that it could be driving is resonances in the molecules themselves. And so there's an idea that the way that proteins find their substrates so quickly is through resonances with those molecules, and that's what attracts them to one another. And then there's other things that are like soliton waves that travel through proteins, and that's what also drives their binding and stuff. It's not just the molecular kinetics. It's also the electromagnetic fields that are being created, and the resonances that are being created in those different molecules.
Yeah, it's exciting. It's like a new biology emerging with all these things we're discovering. It's an exciting time to be studying it for sure. I'd like to switch gears and talk about your paper on signaling as plastic protocognitive systems. And there you describe pathways showing things like habituation, sensitization, and tolerance, where their response depends on prior experience. I found it really interesting. I had not heard about that before. Could you talk a bit about it?
Yeah, that's really cool. So Mike approached me and he's like, "Hey, you want to help write this review paper?" And I was like, "Yeah, okay." And then I was like, "What am I doing? This is, drug tolerance and stuff." And then I started to really dig into it, and I was like, "Whoa, this is fascinating." And basically, they're showing that you can Pavlovian condition any organ that is tied up to the nervous system, and that's any organ. So uh, it's amazing. You can get bile production by Pavlovian conditioning. You can get immune responses by Pavlovian conditioning, and so what's fascinating about that is that we have yet to really tap in to that, right? And so it really kind of gets down to the placebo effect. And, for a long time, the placebo effect was looked at as like, it's just this thing that we have to do in order to, have a good scientific study of how medication is working. But the fact that the placebo effect even exists and that sometimes it's 20% effective, 30% effective is amazing. And I think that that's really what the shamans of way back when, and even now, tapped into, was that idea of belief and how strong the body's the mind is on the body's functions, and what it can do. And so I think that when you prime the mind to be in a state of health, that it can actually have those downstream effects, right? But what's really cool about this is that we've found that cells themselves that are non-neuronal can also be conditioned and can also be taught. And so there's some really great work that looked at Physarum, and they found that, and Physarum is like a slime mold, right? It has no neurons in it at all, and it's kind of interesting because it's like this large cytoplasm with a bunch of nuclei in it. There's not even like individual cells, and so this weird alien slime can be conditioned, and it can anticipate certain things, and so the idea is that memory is stored in even a non-neuronal cell, just through what's going on with, let's say, its cytoskeleton, or what ion channels opened up at that time, or what closed at that time, or what genetically regulated network has been turned on and where it's at kind of in its little circle of dominoes, right? And that those dominoes and where they're currently sitting has an effect on when you come in and push another one, right? And that you don't get the same output every time. And so I really love that because it looks at memory and learning not as something that requires neuronal connection, but that requires an imprint, right? And then you could really just say like a footprint in the sand is a memory, right? And that has an effect on how the water is going to flow over it, and maybe that's going to create another little hill, and it wouldn't do that if it wasn't there. And that all of the physical manifestations of what we do and our actions is like this memory that is constantly building up in the whole. And I think that cells have a certain cyclical signature for how that memory occurs, right? So my husband, Franz Kukling, was working on this, and he's really interested in Bayesian uh, uh, inference, the Bayesian dynamics, and understanding how, uh, in this case, he was looking at algae and how they were reacting to light pulses and whether or not they would have a memory of those light pulses. And he found out that it was due to the rate at which the light enters and then triggers these proteins in the cilia to move. And so if you have patterns of stimulations that uh, either amplify that cycle or dampen that cycle, it will change the way that the organism reacts to it. So that really kind of comes into looking at, hey, let's start looking at our circadian rhythms and what those circadian rhythms will do to reprogram the way our bodies learn, or treatments for cancer, instead of giving the treatment at the same time every day, switching it up, and not allowing the body to get ready to kind of push back on whatever it anticipates is coming. And I found there was a fascinating thing that I learned in that writing that paper was that the majority of heroin overdoses occurs when the heroin user is not in their house, is not in their environment. And it's because they don't have those environmental triggers that then bring up all of the adrenaline and the things that their body is kind of anticipating this thing that's coming. And so then when they don't have that, they don't have the pushback occurring against the drug when it comes in. And so then they're more susceptible to the effects of the drug. So yeah, it's amazing. We need to utilize it more in medicine.
Definitely. Yeah, that was a powerful example. And since reading that paper, I've started looking at some of my own habits and environment and wondering how that might play a role. And I think it's got a lot of untapped potential for sure. But I know you have to get back to an experiment. So I'll ask you one more question in closing. What is something that you're currently interested in or learning about that still feels so astonishing or unresolved that you have a hard time imagining will have a full, I guess, scientific explanation of it in the near future?
Oh boy.
Or just something you're interested in?
Yeah, I mean, I really am interested in the biophotons and understanding that a lot more, um, and kind of disentangling what are all the pulse frequencies that we could potentially be using with this, right? And there's photobiomodulation that is occurring right now. People are using like infrared light to help with wound healing and acne and a bunch of different other things. But what's really difficult with that is that if you get it wrong, you could really cause some damage, right? Because, if you're not using the right frequencies, now you're upsetting like the timing of a lot of different systems. And so I think um, we have jumped in to applying things before we really understand what is actually going on in it. And it's very difficult to study as well, right? And I have constantly been encountering just how difficult a lot of these methods are for reading out voltage, right? We've got really like not very sensitive uh, genetically encoded voltage indicators. So then you have to use dyes, right? And the dyes are more sensitive, but then they have off-target effects. They'll like affect some certain ion channel, or you're trying to get a time-lapse image of cells communicating with one another, but you have to do either really short imaging sessions where you're looking at it the entire time, or you have to do longer ones where you're seeing. And it's kind of like if you have a movie going on and you can only look at so many frames before the movie shuts down. What are the timings for you to understand the entire movie, right? You can watch the first 5 minutes completely, or you can watch, one frame every 2 minutes, or one frame, so it's that same problem, right? And, how to disentangle what's going on and what the language is that people, one thing to have it in English, but like let's say it's in a completely different language, right? Then how do you disentangle it? So what I think I find so fascinating about this subject is that there's a lot of technical difficulty with it, and that's fun. It's fun to try to overcome it and deal with it. And I think the same thing is going to be true with biophoton research. It's very difficult. You have to have particular light sources. You have to have extremely sensitive cameras. You are dealing with electromagnetic pollution all around you. And, what do? You put a Faraday cage around it. Well, Faraday cage is also going to be an artificial environment, right? Because you also need to have what you would, have as an ambiance electromagnetic field. So then you have to apply that artificially. I mean, it gets difficult. Yeah, yeah, it's complicated, but I think fascinating.
Sounds like there's going to be a lot of interesting things coming out of the lab and from you. So I'll look forward to more of that. And hopefully we can have you back on and I'll get you back to your experiment. And I very much appreciate your time today.
All right, thank you.